Okay, can everybody hear me? I think so. That sounds pretty loud. Well, I hope everybody did well on their exam. We're ready to get started into the next section here. We're going to be talking about respiration with regards to lung mechanics, physiology. Here's just an outline of our lectures. This is the generic outline. We will skip around just a little bit as we go through here, but in general, this will be the order in which we go through. One note I want you to make sure of here is our exam here. We will talk about review class. Typically, I would only do review classes during the week. So when we get closer to the exam time, think about what's going to be your optimal review time. And we'll set that up sooner than later. I have a very busy schedule in these weeks here, so if we can come to a consensus sooner, that would be better to make sure we schedule a review. It's assuming everybody prefers a review. All right. Now, this is my office, phone number, email address. Email address is the best way to contact me. So if you need to schedule an appointment for office hours, email me, we'll arrange that, or catch me after class and we'll arrange a time to do so. It's going to be required that you read these chapters in the Byrne-Levy physiology book, chapters 20 to 25 on the respiratory system. I do follow a majority of the material in those chapters. Anything in those chapters that I do not discuss, basically I will not put on the exam. However, it's good for you to look at it. It gives you a broader knowledge and better understanding of some of the material we do cover. So I suggest you read all of this and don't skip over to things that we don't directly discuss because they can be beneficial. I will be using Top Hat Monocle. There will be questions during some of the lectures. It'll be pretty much set up the way Dr. Morales had it as far as the grading goes, the scoring goes. My questions may vary from his questions in the way that they're designed. But nonetheless, we will do that. We will have 2 questions today. We'll do them later in the hour here. It'll be the first time through this for me. Unfortunately, we don't get to practice this and displaying this to the students. What we get, we only get to write the questions. We can't figure out how to actually present them. So if I mess that up, I apologize. We'll get it right the next time. Any questions? Office hours? Schedule? Okay. We're going to be discussing pulmonary physiology in general. This here is just an overview of the pulmonary structure and function. We'll be talking about that first. Then we're going to be talking about ventilation, lung mechanics. Remember, ventilation, in essence, is very simple. You breathe in, you breathe out. You exchange gases as you do so. And this is where the oxygen comes from to supply to all of your tissues in the body. Very simple concept. However, when it comes down to the pure mechanics of ventilation, it's a little more complicated than most people think it is. So it's going to be important that you listen, you understand, and these points of interest are going to build upon each other. So make sure that you understand, and if there's anything that you don't understand, don't be afraid to ask a question. Raise your hand. If I don't see you raise your hand, yell out. I may not see you. If we get too many questions, I may say we'll have to hold those questions till the end of class. But in the meantime, go ahead and ask questions, and we'll go along from there. One thing that you're going to find that I mention in probably every lecture, we're going to be talking about a variety of diseases. I correlate a lot of the things that we talk about directly to diseases. What's the cause of the disease? Why do these diseases come about? What perpetuates these diseases? And, of course, all the characteristics of the various diseases associated with pulmonary physiology. So everything is going to relate to diseases. Now, as we talk about a variety of these different aspects about pulmonary physiology, make sure you can relate these points back together. So we may talk to them broken apart a little bit. I'm going to keep relating them to pure physiology and medical applications. But make sure you understand the big picture on how these things come together. You'll see that when we get toward the end of the section, we'll review some old exam questions, etc. I'll post those for you. But make sure you can relate all of this back to medical application in a variety of diseases. As well, we talk about, within this, diffusion as well, oxygen transport, regulation of ventilation is one of the last things we're going to talk about. All of these things are all going to tie together. And of course, a basic understanding of pulmonary circulation, pulmonary arteries in this case are deoxygenated blood, pulmonary veins, oxygenated blood. Make sure you understand that fundamental when we talk about the pulmonary vasculature, because that is different than systemic, okay? And we're going to discuss that. But you should be pretty much aware and familiar with that before we get started, just to have that basic understanding. This slide you do not have. This just gives you an overview of the functions of the respiratory system. Number one, it's involved in gas exchange. It brings oxygen into the body and removes carbon dioxide from the body. Basic function, most important function of the respiratory system. It's also very important for host defense. This is your primary barrier between the inside of the body and the outside environment. Remember, we're breathing in air from the atmosphere, so whatever's in that air, we're taking into our lungs, into our respiratory system. And the air is full of, whether it's bacteria, viruses, toxins, a variety of different chemicals, gases. All of those are in there, and believe it or not, your lung is a very delicate barrier between the environment and your body. So how your respiratory system handles these molecules and particles that come into your body is very important. We'll discuss that. And thirdly, we're not going to discuss much, but we will touch on it slightly. It's a metabolic organ. It synthesizes and metabolizes many compounds. Many of these compounds are involved in other organ systems, but the lung in general is a filter. All of the blood in your body eventually passes through the lungs quite often. And so therefore, things that are into circulation get filtered out, whether they're hormones produced by other organ systems, other proteins, molecules. The lung is very important as a filter to clear these from your system, and that's where we get a good fine-tuned regulation of your other organ systems. So we're not going to talk about it a lot because we're only talking about lung function specifically here. But make sure you understand that it does clear a lot of those things produced by other organs in your body. Here's some of the diseases that we're going to talk about. This certainly is not all of them, but it's important that you're going to understand these and become familiar with these diseases because we will mention them several times in different respects. So to give you a basic understanding, we talk about emphysema. It involves both the airways and the lung parenchyma. Lung parenchyma is basically the tissue that surrounds your airways and the pulmonary vasculature. So that's all the connection tissue, the integrating tissue. So in that area, there's not a lot of that tissue, but it's very important. And we're going to talk about that, how it's involved in various diseases, one such as emphysema. And this is primarily considered the main cause is smoking. So smoking is your primary cause of emphysema. We're going to talk about smoking being a cause of many problems and difficulties that we find within pulmonary function and pulmonary diseases. Asthma, very common, primarily involves the upper airways. It's also considered reactive airway disease. This is where you just get a constriction of your airways. Many of you are familiar with asthma. We're going to talk about asthma, and we're going to talk about how dangerous asthma really is. It's very well controlled for the most part, but we're going to talk about that very specifically. Pulmonary fibrosis primarily involves lung parenchyma that we just talked about again, and this is primarily due or a response to a variety of different lung injuries, whether it's an exposure to various chemicals, toxins, or even pressure. Okay, we'll talk about barotrauma, which is also a lung injury, and that can induce pulmonary fibrosis. Respiratory distress syndrome, also known as RDS, primarily involves the lung parenchyma as well, and this occurs in premature babies, younger than 32 weeks of gestation. So we're going to talk about respiratory distress syndrome, and that's due to an insufficiency of surfactant. And we'll talk about how the alveolar type 2 cells don't produce enough surfactant, and we end up with RDS. We're going to talk about that in much more detail. Cystic fibrosis. It's a multi-organ disease, but it primarily involves the airways in the lung. This is one of the only genetic diseases that we're going to really talk about, but it's a disease that is very severe in many cases. And so we'll give you a better understanding of what cystic fibrosis is all about. But once again, that is an airway disease. So we're going to talk about these diseases quite often, but we're also going to mention some other diseases as we go along. So make sure you understand what these various diseases are, what their causes are, and what the physiology of these diseases are. They typically become involved in a multitude of my questions on the exam. So first, let's go ahead and get involved with structure and function here. The act of breathing. Primarily, we're going to talk about inspiration. Muscles of inspiration primarily involve the diaphragm. Okay, the diaphragm is the muscle that divides the chest cavity from your abdominal cavity. This muscle, as you can see, it's kind of got this dotted line here. It has a domed characteristic to it. So what happens is when we breathe in, we contract the diaphragm, and when we contract the diaphragm, you can see it better in this image here, the diaphragm, the muscles in the diaphragm shorten, so therefore it tightens, and this diaphragm actually pulls down. So as we shorten the muscles of the diaphragm, this diaphragm pulls down. So what happens when the diaphragm pulls down? We increase the volume of the thorax. Okay? And in doing so, we pull the lungs open and we inhale. So inspiration primarily occurs when we contract the diaphragm. So the diaphragm is a very important inspiratory muscle that we're going to talk about as being the primary factor in inspiration. Now, also very important, which carries a large portion of the load for inspiration, are the external intercostal muscles. These muscles are located between each of your ribs. These external intercostal muscles, once again, during inspiration, you get a contraction of these muscles. When these muscles contract, what it does is it pulls the rib cage up and out. Okay? So the contraction of those muscles, once again, increases the volume in the thorax, pulling the lungs open, helping inspiration. Those are your two major muscles involved, or muscle groups involved in inspiration. It's important to understand that inspiration is considered, in one respect, autonomic, so we don't regulate that. We don't think about breathing. So if you're sitting there sleeping, sitting at rest, inspiration occurs naturally. During that time period, these are primarily the only muscles that are involved. The autonomic nervous system regulates this. There's a feedback system, which we're going to talk about, and how this regulates and is controlled. So that is normal, quiet inspiration. Now, another thing we're going to talk about is a active inspiration, okay? Active inspiration is where we need to increase the inspiratory volume or rate due to primarily exercise or any other cause that causes an increase in oxygen demand. So no matter what activity we're involved in, if we need more oxygen to our body, we're going to increase our inspiratory effort. When we increase our inspiratory effort above and beyond resting inspiration, sometimes we will include other muscles. So in that case, if we need additional inspiratory effort, activation of the parasternal muscles, the scalene muscles, the sternocleidomastoid muscles, these are additional muscles. These muscles here are in your neck, and they're attached to the top edge of your rib cage. And these other muscles, the sternocleidomastoid muscles, these are other muscle groups that also are attached to your ribs along your sternum. They can give additional contraction and opening and pulling up of the rib cage. Okay, so they're only involved when we get into active inspiration as opposed to passive inspiration or quiet inspiration. So it's important you know the difference between those, and make sure you definitely understand that these two muscle groups are primarily involved in all inspiration. Now, when we talk about expiration, it's very important that you understand under normal conditions, as you sit here, expiration is completely passive. And in this term, when we use passive, we mean that there is no muscle involvement. Okay? Expiration occurs with no muscle involvement. As you inhale, you use muscles to expand the lung volume, the chest volume. But when you exhale, it's all recoil. Okay? It's just the rib cage and the diaphragm going, relaxing, and allowing the chest or the volume of the chest wall to collapse. So under normal conditions, expiration does not involve any muscle contraction. So when we talk about muscles of expiration, these muscles are only involved in active expiration. Once again, if we have to increase our oxygen demand, we need to move oxygen in and out of our lungs faster and in a higher volume. Therefore, we have to push the air out in order to bring more fresh air in more quickly. So under those conditions, say exercise, we need to contract the muscles of expiration so that we can further increase our respiratory rate or volume. So the muscles involved here primarily are the intercostal muscles. So the intercostal muscles are opposite of the external intercostals. So these are the internal intercostals. So they work opposite. So when they contract, they actually pull the rib cage down and in, okay, forcing air out of your lungs. As well, we have the abdominal muscles. I'm not going to ask you to understand or remember the variety of different abdominal muscles, the rectus abdominis, the external obliques, the internal obliques, the transversus abdominis. I'm not going to ask you to remember those, but remember it's the abdominal muscle group that's responsible for forced exhalations. Okay? So these muscle groups are only involved in additional expiration, active expiration, or forced expiration, in which we need to increase our respiratory rate and or volume to a higher level other than normal resting level. Okay, so even when you're walking, etc., you typically will not use your expiratory muscles until you engage in exercise that raises your metabolic rate a fair amount. Okay, we can't really put a point on that. It varies from person to person. But under normal conditions, under resting conditions, we do not use our expiratory muscles. So remember, muscles of expiration are passive during normal breathing at rest. Our total lung volume is approximately 4 liters, We're going to talk about how that volume gets broken down and used, and primarily in our next lecture. But it's important to understand these volumes, and we're going to be talking a lot about different volumes. To further go on and talk about the anatomy of the lung, we have the right lung, the left lung. The right lung is broken into 3 lobes. There's an upper lobe, middle lobe, and a lower lobe on the right lung. The left lung only has an upper lobe and a lower lobe. So in essence, the right lung is larger than the left lung. Okay? And that's primarily due because the heart actually takes up space on the left side. Okay? So because the heart takes up space in there, the lung volume on that side, or the left side of the lung, is smaller. Okay? They actually say it's potentially about a 60-40 split. So you can see that the left lung is considerably larger than the right lung. And that's the general breakdown of the lobes. And another important feature here is that we have these fissures. These are divisions between the lobes. There's a horizontal fissure, and down here the oblique fissure. These are between the lobes, and as what that does is it allows those lobes to slide across each other. So when we have expansion, you have a better ability to inflate the lung within those areas. And also, an important clinical aspect of the division of these lobes is that if you have injury or disease in part of your lung, this allows medically them to remove part of your lung more easily. Okay, so they can go in and remove either an injured part of your lung or a diseased part of your lung without affecting the rest of your lung. And then we're also going to talk about how you have a high capacity within your lungs, a lot of extra capacity within your lungs. There's a lot of people, you may have heard, if they have an entire lung removed, they don't notice it much. Okay? Only if they engage in excessive exercise will they notice that, they're missing a lung. Because we have so much extra capacity within our lungs, it protects us against a variety of different diseases. We're going to talk about how these diseases impinge upon our ventilatory ability, but they don't take us down and out until these diseases get extremely severe, because we do have a lot of extra capacity within our lungs to bring in enough oxygen and remove enough CO2 for normal metabolic demands. Okay, so also we're going to talk about an important thing about the lung is the viscera that surrounds the lung. The viscera is divided into 2 major viscera. There's the parietal viscera, and this is actually on the chest wall. So the inside of our chest wall has a small viscera. It's a very thin membrane that covers the chest wall. Also, we have the pleura visceral, and this is the pleura viscera is on the lungs. So we have a small membrane that covers the lungs. So try to envision this, that surrounding these lungs is a small membrane, and then on the inside of the rib cage is a small membrane. Now, between those 2 viscera, this pleural viscera, we have liquid. There's a liquid interface between these. So that small amount of liquid, it's a very small amount, between these 2 layers, and this layer is responsible to allow the lungs to expand and collapse without being abrasive on your rib cage. Okay? So there's a little bit of a fluid layer in there. It's very important. And also this fluid layer is very important for maintaining a negative pressure, which keeps the lungs expanded. We're going to talk about that in much more detail in later lectures. But this space is very important when we talk about it medically. If we remove this space or we open this space to the air, it's called a pneumothorax. In other words, if air gets into that very thin space between these 2 pleural layers. Also, if we have an excess of fluid accumulation within these layers, we get what's called a pleural effusion. That's increased fluid. And also, if we have a lot of infection in here. I believe on your slide this was spelled out as emphysema. It's actually empyema. Okay, so that's different than emphysema. This is actually where we have an infection within this layer. And when we talk about nerves, pain, there are no pain receptors within the lung other than in these pleural viscera and the parietal viscera. So within the pleura, there is pain receptors. So basically, as a clinician, if somebody complains about excessive pain within this region, it can either be due to a pneumothorax, pleural effusion, or an infection. So there you can actually sense pain. And within the rest of the lung, there are no pain sensory neurons. And down here, we're not going to discuss it a lot. It's just to understand your basic anatomy. When we're talking about apical, we're talking about the top of the lung. Basal, we're talking about the bottom of the lung. And make sure you understand what, when we're referring to anterior, posterior, lateral, okay? These are just basic anatomy terms to give you definition, whether we're talking about the lung on this surface, the back surface, the side, okay? Just make sure you have a general understanding of these anatomical terms. We will not discuss them very much in detail, but they are helpful for you when referring to very specific areas. But make sure you definitely do understand one, apical and basal, because we're going to talk about distribution of airflow and blood flow with an apical and basal distribution. So make sure you do understand at least those. Now we talk more about the airway. So as we talk about the airway, the upper airway has these cartilage rings that surround it. As you can see, the airway branches down into multiple branches. And you can see we've got the right, and we've got the left. They're similar, but yet you can see here we have the three branches on the right side, the upper lobe, the middle lobe, and then the lower lobe. Okay, as opposed to the left, which just has the upper and the lower. Okay, there is no middle lobe. So this small little middle lobe here is the only thing that's much different between the two sides. But the important part here is what we want to demonstrate is that the larger airways have cartilage rings around them. The importance of the cartilage rings is that it prevents those parts of the airway from collapsing easily. Okay? These cartilage rings go about 80% around the airway, so there is still room for constriction and relaxation of these airways. And of course, as we talk about asthma and some of the other upper airway diseases where you can get constriction, that's very important. So even though these cartilage rings help to prevent full collapse, we can still get a narrowing of these airways. So you can see the branching structure here, and we're going to talk about that in more detail. Here's a cast of the airway, and in this cast you can see all the parenchyma has been removed, so you can see all the airway here, and that's in the white. And you can see how finely this branches out, just like a tree. You know, it goes out to branches upon branches and bifurcations. And also there's a partial cast of the pulmonary arteries and pulmonary veins, which you can see here in the red and the blue, very lightly in this sense. So it shows the relationship between the blood vessels and the airways. We're going to talk about that in much more depth, but this is just to give you a bigger picture. Here we talk more specifically about the upper airway, in which we call that conducting airway. The conducting airway is the airway that primarily has cartilage around it. And we start with the bronchi. Those are the larger airways. And then we go to the non-respiratory bronchioles. Now, as we get into the bronchioles, once again, we have cartilage in some of the upper bronchioles. But as we get down to what's called the terminal bronchioles, here we do not have cartilage associated with these, but these airways, the terminal bronchioles, do not get involved in gas exchange. So the next unit we refer to here is the respiratory unit. As we go further down this branch, these portions are involved in gas exchange. So the difference between conducting airways and respiratory units is that the respiratory units or the respiratory airways are involved in gas exchange. So there we have the respiratory bronchioles and the alveolar ducts and the alveoli. Now the conducting airways have a dead space of about 150 ml. Can't quite see that, but that's 150. That's 150 ml. So if we look at the volume of the conducting airways, that's about 150 ml. And a normal tidal volume is about 500 ml for a normal-sized adult. So with respect to your total tidal volume, 150 ml of that 500 goes into this conducting airway, and that is not involved in gas exchange. These numbers are going to become very important when we talk about alveolar ventilation and oxygenation. So keep that in mind. Now, the respiratory bronchioles and the alveoli, they have about a volume of 250 ml. This is their capacity. Now remember, when we talk about a tidal volume of 500 ml, what does that tell you? If their normal volume is about 2,500 ml and a tidal volume is 500 ml, that means we retain a lot of gas or a lot of air within our lungs with each breath. Okay, so we're breathing on top of a volume that's maintained there. Once again, that's going to be very important when we talk about gas exchange. Another important thing here is that when we talk about surface area, the surface area of the respiratory units is about 70 square meters. Okay, 70 square meters is approximately the size of a tennis court. So if we were to take our respiratory portion of our lung and spread it all out, it would cover a tennis court. That is a huge amount of surface area. This surface area is very important, once again, when we talk about gas exchange. Surface area is directly proportional to the amount of gas or oxygen and CO2 that we can exchange between the airway and the circulatory system. So keep in mind that this large surface area is very important, especially when we talk about the disease of emphysema. We're going to see how the decrease in that surface area can be quite dramatic. So here, once again, you see as we branch down to here we go down to like 23 branches. So we have a lot of bifurcations. But make sure you understand that difference between the respiratory airway and the conducting airways. Here we look at it in another respect. So once again, here's our branching airway. We have our conducting airway of about 150 ml. Now that's called anatomic dead space. So remember that term. We're going to talk about it again. But the anatomic dead space refers to the gas volume that's in the conducting airway that is not involved in gas exchange. And then we have our exchange surfaces here. This talks about the divisions. The important matter here is the diameter. As you can see, the diameter of these branches decreases dramatically as we move down. Anywhere from 15 to 22 millimeters at the trachea, all the way down to 0.3 millimeters of the alveoli. So you can see that the diameter gets extremely small as we come down to these branches, especially in the air exchange surfaces. These are extremely small diameter airways. The important factor to go along with this is how many of these branches are there within the lung. As you can see, there's only one trachea. And so if we actually measure cross-sectional area at each level, you can see the trachea has a 2.5 square centimeters. But as we go down the respiratory tree, the number of these branches increases exponentially. Therefore, the cross-sectional area associated with how many there are. So we talk, they're smaller, but there's more of them. But the important thing is the cross-sectional area increases. So as we get all the way down into the alveoli and we talk about total cross-sectional area, we're greater than 1 million square centimeters relative to the 2.5 square centimeters at the trachea. So when we look at this and you think about as we're breathing in and we're moving air into our lungs from the atmosphere, what happens to our airflow? So here we talk about airflow velocity. So velocity is going to be highest where the cross-sectional area is the smallest. Okay? And as we increase the total cross-sectional area to very large, well, we have the same volume of air moving, but now the velocity is much slower because we have a much greater cross-sectional area. Make sure you understand this relationship. So airflow velocity decreases as we go distally in the lung. Now, if we compare that to resistance, okay, we're talking physics here, so don't get lost. We're talking resistance to airflow. It also goes the same way. If our cross-sectional area is small, we have a high resistance. If we increase that cross-sectional area, we decrease resistance to airflow. So resistance to airflow also decreases as we go distally in the lung. Very important concepts that you need to remember. We're going to talk about airway resistance and how important the changes in airway resistance are related to disease and the ability or inability for us to move air efficiently from the environment into our lungs. Okay? So once again, we go back and we're going to talk about lung diseases continuously. We talk about lung diseases of the upper airway. Now, more specifically, this is a group of diseases known as chronic obstructive pulmonary disease, or COPD. This involves primarily just the conducting airways. As we mentioned, some of these, asthma can be involved with inflammation and also airway smooth muscle constriction. So basically, asthma is caused by a narrowing of those upper airways due primarily to constriction of the airway or inflammation. Bronchiolitis, primarily found in young infants, involves the smaller airways, but it's primarily caused by RSV. Okay? That's a virus that's associated or found primarily in younger infants. So RSV is a respiratory syncytial virus, and that actually causes an inflammatory response as well, but it's due to a virus. So that is considered to be called bronchiolitis. Once again, it's a narrowing of the airways due to an inflammation from the virus. Chronic bronchitis, you hear about people with chronic bronchitis. It's quite common, primarily found in smokers, once again. And this is a narrowing of the airways primarily due to an increase in mucus secretions. Okay, so we get more mucus secretions in the airway, creating a narrowing because of the mucus secretions, and also increases resistance to airflow. And that's a chronic bronchitis. We're going to talk more specifically why smokers have chronic bronchitis and how that affects the mucus secretions. So as you can see, all of these so far are primarily a narrowing of the airways, but due to different causes. Cystic fibrosis, we mentioned, is a genetic disease. It affects a very specific chloride channel, movement of fluid. We're going to talk about this in more detail, and that's also an obstruction by mucus. Once again, mucus, very similar to chronic bronchitis, but a completely different cause. In this case, it's a genetic disease as opposed to that induced by smoking and various things that we breathe into our lungs. So these affect the upper airway, the conducting airway, and they're called chronic obstructive pulmonary diseases. So now if we look more at these micrographs, we can look at this a little more in detail. This here you can see is a large conducting airway. You can't see it very well here, but the large conducting airways are covered with cilia and also mucus. There's a lot of glands that secrete mucus into the upper airways. This would be mucus on the surface of the cilia. And then next to this here, we have some of the alveoli. The alveoli are much smaller in diameter, and you can see all these very small pores within the alveoli, and that's because all these alveoli are pretty much connected to each other. We're going to discuss that and how important that is, because that helps to inflate these alveoli optimally and uniformly. Here's a magnified picture, once again looking at the conducting airway. These are ciliated epithelia. You can see all these cilia. And all these bigger round spots you see in here are called goblet cells. Those are the cells that secrete the mucus into the airway. So you can see that there's a high number of these goblet cells, and that your entire surface of your airway in a conducting airway is covered with cilia. And of course, the cilia is responsible for moving things that we don't want in our lungs up and out of our lungs. We're going to talk about that in more detail as well. But it's kind of, this is something that you don't think about. You think of your airways as being very clear and bare and don't really have much on them, but they really are all covered with these cilia and these other cells secreting mucus. Once again, a higher magnification. You can see these cilia. It kind of looks like a carpet, inside your airway. And of course, these are mucus goblets coming out from the goblet cells. So this is mucus being secreted. The goblet cells kind of sit down within the cilia, kind of below them, so you can't really see them, but you can see the mucus as it comes out through the cilia. And there we are. That's just an opening of one of the goblet cells. Typically, this is all covered with cilia, so you can't really see that. But this mucus comes out in these balls of mucus, and it comes up to the surface, to the top of the cilia. And we're going to talk about more of that and how that actually functions, because there's other cells that secrete a fluid. The epithelial cells secrete a fluid within the airway as well, and mucus kind of floats to the surface of this liquid. And that's going to be very important. We're going to talk about that, especially when we talk about cystic fibrosis, as well as chronic bronchitis due to smoking. We're going to talk about how that is altered. And this here just represents, once again, this is the lumen of the airway. These are epithelial cells. Okay, epithelial cells cover all surfaces that come in contact with the environment. Okay, you have epithelial cells on your skin, in your airway, in your GI tract. Believe it or not, your GI tract basically comes in contact with the environment because everything you eat comes from the environment and passes through. So anything that has contact with some sort of the environment is an epithelial cell versus an endothelium cell. Endothelium is the endothelial cells that line the blood vasculature and also the lymphatic system. So those are endothelial cells. We're going to talk about, when we talk about respiratory physiology, the important thing is that the epithelial cells, which are in contact with the air surface, are very close proximity to the endothelial cells, which are in contact with the blood. We're going to talk about that very close contact and how important that is for gas exchange. So now we go back to the structure of the cilia. Here the cilia is the cilia cross-section. There's 9 doublets of this actin and ATPase. So this is one single cilia hair, and you can see all these different actin and ATPases. And in the center, there's 2 single units. Now, these are comprised of a dynein arm and a nexin link, and they're all attached to these in the center. It's important to understand that these consume ATP or energy. Okay, so it takes energy to move these cilia. The cilia move in a very designed fashion, and that movement is because of this structure within the cilia. So all cilia move directional, even though they go back and forth, but they go back and forth within a manner in which they're able to move fluid in one direction. In this case, cilia moves fluid from distally to proximally. So they bring fluid out of your lungs, okay? And that's helped to keep the lungs clear of any types of infection or any particles or molecules that get in there. Your airway actually has good defense and the ability to remove bacteria, pollen, dust particles, anything that gets in, as well as viruses. So viruses are very small, but even those can get captured within the mucus and the liquid layer, and they're removed from your airways. So you have a very good host defense against anything that's in the environment from getting in. Also within your lung system, as we're talking about host defense, we talk about alveolar macrophages. Now, the macrophages migrate in from the bloodstream. We talked about there's actually a lot of blood flow that comes through the lungs. The blood is in very close proximity to the epithelium, which is in the surface of your airways. So the macrophages migrate through from the blood into those air spaces. So as they go into the air spaces, they scavenge dust particles, bacteria, viruses, and once again, the cilia move the macrophages and all that they ingest up and out of your airway. So the macrophages are very important because not only do they take care of things on the surface that get caught within the airway, but they also can migrate through and they can be deep down at the tissue level or along the cell level. So they can really they act as little vacuum cleaners basically, and they mop up everything that gets in there. So now we're going to talk a little more about the innervation. I don't like to go into a lot of depth about innervation of the respiratory system, but we're going to talk about the basics. This diagram here is about as much detail as I prefer to go into. We'll talk about it just a little bit more. But primarily, what we're going to talk about is in the autonomic nervous system, we're talking about the parasympathetic system. Now, the parasympathetic system starts here in the medulla, and it comes down through, and primarily we're referring to the vagus nerve. The vagus nerve is your primary pathway of the parasympathetic system. Parasympathetic system uses a neurotransmitter of acetylcholine, and primarily the parasympathetic system causes airway constriction, blood vessel dilation, and glandular secretion. Okay? It's important just to remember those three things that occur from the parasympathetic system, and remember that that's primarily through the vagus, and that comes down and then branches and innervates the lung as well as the blood vessels and the glands within the lung. And we'll see some other pictures that kind of show that innervation, but make sure you understand this is the major functions of the parasympathetic system, as well as the sympathetic system primarily secretes norepinephrine or epinephrine. This is the neurotransmitter, and this pretty much comes in here off of the spinal cord, innervation along the spinal cord, and this comes out and innervates once again the airway. It causes a relaxation of the airway and blood vessel constriction, as well as inhibits glandular secretion. So this is opposite of the parasympathetic system. So make sure you know the difference between these two. And remember that these innervations go to these parts of the respiratory system, and it's a summation of the inputs. So the parasympathetic system and the sympathetic system are always active. They're both always active at various times, some greater than other times, but it's a summation of these activations or inhibitions that is the result of what happens. So Obviously, if we have a higher parasympathetic activation, then these things occur as far as airway constriction. So they're always in competition. There's always a balance. And we previously mentioned that there's no pain fibers in the lung, only in the pleura. So there are no other pain fibers associated with this. Now, another thing I want to point out here is these motor neurons or these skeletal neurons. Okay? If you look at the innervation here associated with the spinal column, you can see that this is the phrenic nerve. The phrenic nerve innervates the diaphragm. Okay, we talked about the diaphragm, how that's very important in inspiration. So without the phrenic nerve, our diaphragm would not function, and we would not be able to use that for inspiration. And also the intercostal nerves, those come off of the thoracic innervation here, and you see how that affects the intercostals. Therefore, the external intercostals that are responsible for inhalation are also controlled here. Now, if we have injury, a spinal injury, make sure you understand that depending on where that spinal injury occurs, what effect is that going to have on our ability to inspire? Remember, expiration is passive for the most part. We don't have to worry about that. But as far as inspiration goes, so anybody who has a very high spinal injury, say C1 or C2, what's going to happen to their ability to breathe? Going to be completely lost. Okay? So the only way that they'll be able to breathe is through mechanical ventilation. Okay? And I'm sure you can think of different examples of people who have been injured with upper spinal injuries where they have to be on a ventilator all the time because they cannot breathe. So they need mechanical ventilation. Now, if you have a spinal injury down at C6, C7, you're going to lose your ability to use your diaphragm. You're going to lose your ability to use your external intercostals, but you still have the ability to use your diaphragm. Diaphragm, like I mentioned before, it's actually about 60% of your inspiration. So you still have the ability to breathe, inspire to a large extent. Now, the lower that injury, the less you lose as far as ability to inspire. Okay, so it's very important that you can see that and what happens. Now, when we talk about the sympathetic system here, you can see how these nerves all come together. They're actually all tied together through these ganglia. So no matter where that injury occurs, we're still going to have some effect from the sympathetic system. So we do not interrupt that dramatically. As well, obviously, any injury we're not going to have in the medulla, so that would be more of a spinal or a brainstem injury. So we don't have to worry about this type of innervation in injury, but we do worry about the motor neurons and the ability for the inspiratory muscles when we talk about spinal injuries. Okay, so this kind of ties a little more of this together. We talked about parasympathetic preganglionic innervation. This here is your spinal column. This is some of the ganglia that we have, as well as the sympathetic postganglionic innervation. And we see that the innervation all comes in. It's all summated. We have excitatory inputs. We have inhibitory inputs. Some of these neurotransmitters are a little different depending on where they're located. We're not going to be as concerned about some of these other neurotransmitters other than acetylcholine and norepinephrine. So we're going to just primarily focus on that, but you can see how the innervation from the sympathetic and parasympathetic systems all come in and innervate here, in this case, the smooth muscle system, the glands, control of secretion. You can see how we have parasympathetic and sympathetic. And also the airways. Once again, that's a smooth muscle that causes constriction. And we don't show any vasculature here, but they also affect the vasculature. You get innervation from both systems. Other than that, there are some very specialized cells. This is the Kolchitsky cell. It's not really known for sure what its function is, but it is known that it's a neuroendocrine. It secretes dopamine and serotonin. Other than that, we don't have a real good function for that cell. Other than that, it is found within the lung epithelium. Okay, so this kind of just gives you an idea where everything ties in together. Once again, this is cartilage associated with some of the upper airway, but there's still smooth muscle cells or muscle cells associated with this that can give us a constriction of those airways. So here is just, once again, just a summary of the control system or regulatory system associated with the respiration. We have our cerebral cortex. We have our respiratory centers in the medulla. We get nerve impulses through the spinal cord, respiratory muscles. And there's also mechanoreceptors in the lung and the chest wall. These help to feed back and tell us how much volume we're taking in. Okay, so it's very important that these mechanoreceptors come into play. As well, as we go down through, we look at our alveolar capillary barrier, in other words, oxygen and gas diffusion, how much oxygen is being moved, how much CO2 is being moved, what's the pH of the blood. There are chemoreceptors associated with the blood flow and these oxygen and CO2 levels that also feed back as chemoreceptors and help to regulate these nerve impulses to say, how much inspiration do we need? How often do we need to inspire? So that all controls our inspiratory and expiratory efforts. So this just gives you the bigger picture of these feedback loops. We're going to talk about them much more specifically in later lectures. And so once again, here's a summary, kind of pulling it all together in a picture. And in this picture, you can see how alveolar macrophages, even though they migrate through these cell layers, they can effectively move along these surfaces. Ultimately, they end up on the outer surface or in the airway. This is the air lumen here. So these macrophages continue to pick up. Now here, the important thing is these are the cilia that you see in the ciliated epithelial cells in the conducting airway. Now remember, when we get to the respiratory airway where we have gas exchange, there are no cilia. Okay, so the cilia are only found in the upper airway. So here, this is called a sol layer. This is solution. Okay, and we're going to talk about this in more detail when we talk about cystic fibrosis. But here you can see this is a solution. So these epithelial cells secrete a fluid into the airway to maintain this solution here. This is like a saline solution. It's a sodium chloride solution. And it allows the cilia to move freely. Okay, so it's just a water solution. And the mucus that is secreted from these goblet cells, quickly as it's secreted, finds its way to the surface of this sol solution. And it will basically sit on top of the fluid layer. Now, the mucus is very sticky. So basically anything that comes in through the air, dust, bacteria, viruses, if it comes in contact with this mucus, it sticks to it. And as it sticks to it, that also helps to immobilize it, allowing macrophages to engulf it, as well as allows it to be moved up and out of your airway. So you can see the importance here of how the mucus comes into play, and it sits on top of this cilia or this solution layer. Now, when we talked about smoking, there are components in smoke that inhibit cilia movement. Okay, so it basically slows them down. I'm trying to remember. Here we can see that cilia normally moves about 1,000 times per minute. If you're a smoker, it slows it down to about 20 times per minute. Okay, so now you can see the importance of how smoking causes a chronic bronchitis. Because now we do not have this moving out of the airway. We get an accumulation of this mucus sitting on top of this fluid, and everything that's sticking to it it moves out of the airway much slower. So therefore, smoking inhibits the cilia motion, and we get an accumulation. I'm not a smoker, so I don't have chronic bronchitis, but so you can see what happens. And when they talk about smokers' cough, and this is why. Smokers have a cough quite often because they have to cough to facilitate the movement of this out of their airways. Okay, especially early morning when they've been sleeping, this even accumulates a little more. Okay, and also in smoking, you get a hyperplasia of the goblet cells. All that means is the goblet cells get bigger, they secrete more mucus, so it's kind of a double-edged sword. Not only does it slow down the cilia, but they secrete more mucus, so now you get more plugging and narrowing of your airways. So smoking is not a very good thing as far as cilia motion and moving things out of your airways. We're going to talk about other effects of smoking, especially when we talk about emphysema. Here, we're going to talk about just a little bit about cystic fibrosis. Cystic fibrosis causes a very similar issue relative to the movement of cilia and the mucus up out of the airway. It's a genetic disease in what they call the CFTR gene. What happens is it's a chloride channel that becomes dysfunctional. In order to get this sodium chloride solution out here, the chloride channel has to move chloride into this space. If we don't get the movement of chloride, we don't get the movement of sodium associated with that, and then we don't get the movement of water. Therefore, is what happens in cystic fibrosis. Question?
I have a question about the smoking part.
Sure.
Are those effects just from cigarette smoking, or is it any type of smoke that can do that?
Oh, any kind of smoke. Cigarette smoke, it has thousands of different components within it. Regular smoke from, say, a building fire or trash fire, burning leaves, etc., also has a large number of particles, etc., in it. So even that type of smoke will have effects to it. So the smoke composition can be a little different, but it's also a little different whether you smoke cigarettes or cigars. So, but they all smoke in of itself has so many different molecules within it that create this problem. So smoke in general will cause this. So back to cystic fibrosis, we have an inhibition of the production of this solution layer. So as what happens is, as this solution layer disappears, this mucus layer up here settles down into the cilia. So now the cilia are caught in the gel layer here. So that gel layer is very thick. And as you can see, if it's very thick, it's going to inhibit the cilia motion. Okay, so the cilia can't move very well when there's all this mucus is stuck within the cilia. So that's what cystic fibrosis is. You get a similar effect. You're unable to move this mucus up and out of your airways. So people with cystic fibrosis quite often have to be suctioned on a regular basis. They actually put a saline solution down into their airway, and then they take a suction tube and suck it back out just to try and keep those airways clear. We're going to talk about that a little more in cystic fibrosis. There's multiple genes that can be affected, and depending on which genes are affected, the severity of cystic fibrosis can vary. Some are very severe and some are mild. So you'll have varying degrees of cystic fibrosis in which that causes the inability of you to move this mucus layer out of your airway. Okay, and once again, we talk about our innervation here, the cross innervation between the sympathetic nervous system and the parasympathetic nervous system, whether it's on the mucus glands or on the muscles. So just make sure you understand that in general, that relationship. So here we talk about cystic fibrosis specifically. The occurrence is 1 per 2,500 births in whites, and it changes depending on what your ethnicity is. It's even rare in Asians. 5% are carriers of the recessive gene defect. It's on chromosome number 7. And then it goes on and on, talking about the incidence of cystic fibrosis. And the CFTR gene is the cystic fibrosis transmembrane regulator. Okay, and that encodes for the protein that helps for chloride transport. There are over 300 CF gene mutations. So there's a wide variety of different mutations that can occur, and sometimes you can have one or more of these deletions. And each defect compromises airway clearance to different degrees, as I mentioned. This is the most common defect. It's at position 508. It's a phenylalanine deletion. So make sure you understand what cystic fibrosis is, that it affects a chloride channel, and how it affects the movement of mucus in this layer out. Different ways of treating and can help restore function in mucus layer and airways includes increased chloride secretion with uridine triphosphate. So pharmacologically, they can help that secretion of chloride. They can decrease the sodium absorption with amiloride. So once again, trying to get sodium within there. So usually sodium reabsorbs at different levels. And also decrease viscosity of the music with, like I said, there's a Dornase alpha, and that cleaves the mucus proteins. That's just to break up the mucus in the airway. They can use that in conjunction with the saline that they put into the airway to help move that out along with suctioning. And of course, inhalation of the saline solutions to enhance ciliary function. So make sure you understand that, what cystic fibrosis is and how it occurs and what happens. So now as we talk a little more down in toward instead of the conducting zone, we're talking about the respiratory zone. Here we talk of the bronchioles, no cartilage. This is the smooth muscle cells that surround the epithelial cells. And if there's no cartilage, so now if we get a constriction or a relaxation of this smooth muscle layer, we can get a narrowing or relaxation of these airways. And you can see that we can affect airway resistance dramatically, especially down in the lower bronchioles where there is no cartilage. Here's just a pulmonary artery. Once again, you can see the close proximity of the airway with the blood vasculature. Very important. We're going to talk about that. But these are large vessels, so this is not where our gas exchange occurs. Our gas exchange occurs primarily in these alveoli, which you can see out here. These are all air spaces, and these are all blood vessels associated in and around these air spaces. So if you look at that really closely, you can see that's where our gas exchange really occurs. But these are the bronchioles. These are actually considered larger diameter within the lung compared to some of these alveolar diameters. And we skip back just a little bit for a second here. This is one of the axon varicosities, or this is like the vagus nerve, or a nerve that comes across the smooth muscle cells. So these are muscle cells underneath here. As this nerve comes across here, these axon varicosities, this is where acetylcholine is released in little packets. And what it does, it comes out and it kind of rains down over the muscle and causes constriction or relaxation, depending on if the muscle is associated with airway or vasculature. So this is just an example of how these neurotransmitters actually affect the muscle layer. So here, if we're talking about acetylcholine, we're talking about parasympathetic through the vagus nerve. I'm not going to ask you about the various receptors associated with the different innervations. In this case, it's a muscarinic receptor, subtype 3, that's associated with acetylcholine. Skeletal muscle innervation, those have nicotinic receptor, subtype 1. Like I said, I'm not going to talk about the receptors, but different organ systems or different muscle cells have different receptors that respond differently to the different neurotransmitters. So it's important to know that they do have different receptors and they have different effects based on that. But I'm not going to specifically ask you which receptors are found in which tissues. And this is just a cross-section looking at it in a different direction. These are some of the vesicles containing acetylcholine. So as they come out and they're released, then it releases the acetylcholine in the proximity of where the varicosities are. And this here is just a experimental image here. So in its controls, what we're looking at here is these smaller airways here, and we're going to look at the effects of constriction on these airways. So if you look at these airways here, and now we're talking about, we're going to stimulate acetylcholine to be released, and we're going to get airway contraction. So here, through vagal stimulation, and there's different ways for vagal stimulation, you can do that either here in the neck. You can actually physically stimulate the vagus, or you can pharmacologically stimulate the vagus. Either way, you get an acetylcholine release, and you can see we get a constriction of these bronchioles. And we can actually get up to a 30-fold narrowing of these airways. Okay? So the cross-sectional area can be a 30-fold decrease with this activation. So these airways are very responsive, and it's very important to understand that, especially when we're going to talk about asthma, how critical that can be. And here we go. We're going to talk about just another aspect here when we're talking about asthma. There's a variety of different ways we can treat asthma. If you notice, most asthmatics carry an inhaler with them. In that inhaler, it's basically falls under the broad category of a bronchodilator. These bronchodilators can be one of many different things. Here's an example of 6 different things that could be found in these asthmatic inhalers. In albuterol, it's a beta-2 adrenergic receptor activator. Or you can use atropine, which is an M3 inhibitor. You can use steroids, which inhibit inflammation. Antihistamines, which affect the mast cells. Mast cells are found within the airway that secrete histamine. Histamine causes constriction, so an antihistamine. Leukotriene in combination with steroids, which is found in Singular, and VIP, which is another neurotransmitter, which can cause a relaxation of the muscles. So depending on the type of asthma that a person has, or the sensitivity or severity of that asthma, the doctor will prescribe a variety of different inhalers to optimize the response and the ability to have dilation. Now remember, if we can have a 30-fold constriction of these airways, we can make it extremely difficult to breathe during an asthma attack. So it's very important that these inhalers are used immediately. And we're going to talk about it later on, what happens if you don't use these inhalers immediately, how as you increase airway resistance, it actually cycles back and causes further constriction of the airways. So it compounds, and it compounds quickly. So the problem is, if you don't use this inhaler at the first signs of a problem, how does this inhaler work? The inhaler works by getting this pharmacological agent into the airway to the site of the constriction. Now, if you have too much constriction, what happens to your volume of air that you can bring in? Decreases exponentially. So now if you can't breathe in enough air to get that inhaler compound into your airway, you can't affect it. So what happens is, asthmatics, and you'll notice this, somebody, taking a puff on their inhaler, you don't notice that they have difficulty breathing. But the person themselves, the first sign of difficulty, which to them they notice it much quicker than somebody else would, they're taking a puff on that inhaler, because prevention or early treatment is the only way that they can treat it. Because otherwise, if they get constriction, it creates a further constriction. And we're going to talk about that later, how that cycle occurs and how quickly that can occur. So just as a point of interest, people who are always taking a puff on their inhaler, even though you don't notice any major difficulties, it's important that they do take that very early. Now, we talked about the lungs being important in medicine, the ability to deliver drugs. Now, when we talk about inhalers, we're administering the drugs directly within the airway, but also it's been known and research that we can deliver a variety of drugs through the airway. We mentioned already how the close proximity of the airways are to the blood system when we get down to the alveoli level. So if we can administer drugs in an aerosol that we can get deep deposition with, we can actually get that drug to move through the lung epithelium into the blood system. So the lung is a very effective delivery system for a variety of different drugs. And here's just an example of an article that talks about some of the different drugs that are now available as delivery through the lungs. And it also talks about some of the possibilities of inhalation therapy for such things as delivering insulin for diabetes. We actually did a small trial here at the university where we were looking at different ways that they can formulate insulin in a dry powder that can be inhaled through an inhaler so that you can get effects that way, as opposed to taking insulin shots. So once again, if you have a further interest, you can look at that and how the airway is very effective in delivering certain types of medications as well. So as we talk about further, we talk about the respiratory or the gas exchange areas. This here we're looking at the alveoli. This here is a pulmonary vein. This is a terminal bronchiole, respiratory bronchiole. Remember how we said the terminal bronchiole comes down and there's no cartilage, but yet it is getting close to involved in the gas exchange area. So this here you can actually see how the terminal bronchiole turns into the respiratory bronchioles, and it's very closely associated with the gas exchange areas. And this here is an alveolar duct. This is a pulmonary artery. And of course, if we look at this in a higher magnification, we can see how the macrophages are found down in these alveoli, once again, moving around, picking up any particles or any material that should not be there. Once again, preventing any types of infection or disease. And here, this is a blood vessel, and these are red blood cells. These red blood cells actually have to turn sideways because these vessels are so small, that's the only way they can get through. But remember, this is an air space. This is a blood vessel. Look at the close proximity between the air space and the blood vessel, once again, for optimizing gas exchange. And we're going to talk about that distance. Here's just a size here. This is a 10 micron scale bar. Typically, a red blood cell's a little smaller than 10 microns. So you can see how small these blood vessels really are in the close proximity. So once again, here we are looking at a bigger picture, the terminal bronchiole, respiratory bronchioles, the alveolar surface. Once again, we say that's got a surface area size of a tennis court. And the other important thing to go along with that is the capillary blood vessels associated with the alveoli cover about 96% of that area. So we have an air surface the size of a tennis court, but we also have a blood volume relationship with that air surface that covers about 96% of it. So that huge surface is in close proximity to the vasculature, so we can facilitate gas exchange, and that just demonstrates the size of that area. But this whole capillary blood volume is only 60 ml. So can you imagine taking 60 ml of blood and spreading it over a tennis court? That's how thin that layer of blood would be, or as we demonstrated, those blood vessels and the red blood cells are just squeezing through and coming into close proximity. So that just wants to demonstrate the magnitude of this close approximation. Here we talk about terminal and respiratory bronchioles, but I guess now is what we'll do is we'll try and do our little test questions if you guys are ready for that. So we'll give you a minute to do that. Oops, there we go. That's what you need to see. Oops. Now, unfortunately, like I said, to activate that, I must have... Okay, I guess it's going. I'm sorry, the clock isn't... There it is. That's submissions. I have to refresh on how to bring this up. Unfortunately, I can't look at this in my office because it doesn't allow us to do that. We don't have access to this software that's on this computer. So anyway, go ahead and pick the right question or answer. Which of the following diseases is primarily caused by the insufficient production of surfactant? We talked about that earlier in the lecture. It's not showing up? Okay, we'll try this again on Thursday. Like I said, I think I'm probably missing something here because it's not coming up the way it should have. So what we'll do is we'll pick this up from here on Thursday, and we'll continue on. Thank you. You know, I don't know which clock to go by these days. Okay. That one's wrong. Is it slow or fast?
It's slow.
Oh.
I have a quick question. You said that for asthma it's better to take it right as soon as we